Welcome to the Rahbar Talk Show. This is our second session related to monitoring the cancer biology. And we want to highlight today about the different diagnostic tools, how we can just detect the cancer cell at early stages, even that to prove the occurrence of the cancers and neoplastic condition. So be with us and see what happened. Okay, Mrs. Ojari, as you know, under different circumstances, cancer patients referred to the hospital and clinics are at the late stage of their own condition. You know what I mean? I mean that the tumor cell distribute to the different organs, and even the size of tumors increased. And so, do you think that the early stage detection of the tumor cell can help to increase their survival rate or not?
Okay, first of all, I want to bring a summary from our previous session. We said that cancer is a condition when we have an uncontrolled cell division and normal conditions. We have genetic or molecular factors that control these extra proliferation. Even the apoptosis of the cells are controlled by these factors. As a cancer develop, we see multi-factors in cancer. It can cause by one of them or all of them together. The mutations in cancer suppressor genes like P53 or RB, or the mutations in oncogenes. We can see all of them in cancer cells. But I have read some articles that they say that the early detection can help this condition, because I have some reasons for that. I want to bring two important of them. First of all, where in the early stages, the cancer is localized in a specific tissue or organ, and it hasn't get that time to spread surrounding tissues, or or they haven't the cancer hasn't gone to the.
You mean that have a metastasis to the different sites?
Yes, to the distant organs, when we call it metastasis. So when it hasn't, and about the immune system, they haven't thought to escape from the immune system that much. And when we use for example, a treatment, like we can remove them by a surgery, or we can control them by radiotherapy or chemotherapy.
But sorry, I'm going to interrupt you. In most circumstances, we have no solid tumors. I mean that a solid mass can be detected by surgical approaches. Even that, for example, in the early stages, we could not, see the tumor mass on gross view, and so we could not just excise it. And so, how it can be possible? So we just give a chance to tumor cells to just increase. Is it right or not?
I'm talking generally about the most solid tumors. I'm saying that in the first stages.
I'm going to just define it precisely. You mentioned early stage. Early stage, what does it mean exactly? For example, in the case of the blood tumors, it can be possible. But in the case of the solid tumor, I think it is actually impossible.
But some of them, when go to the hospital or and they visit the doctor, they have some symptoms. I think, for example, they have abnormal pains or they have.
Yeah, we call it a neoplastic syndrome. These are some the symptoms related to the occurrence of cancer in each individual. So we have some, I don't know, the clinical manifestation like a fever, I don't know, the cachexia, something like that, emaciation, something like that. Okay?
But maybe you're right. We cannot see the tumor, or we cannot detect that tumor from the early stage in all cancers. But generally, if we had that possibility, I'm saying that detecting in early stages is better, because the tumor hasn't, gets through the metastases, or
we just have seized the
yes.
metastasis from the one side to another side and formation of perimetastatic foci in ectopic site. So thanks a lot. Great.
Dear Professor, I want to bring the second reason about why we have to why the early detection is better. The second reason is that as the tumor start to proliferation,
advances and progresses.
Yes.
Yeah.
We see different mutations. One of them is that the structure of the DNA is getting more different and different, as in when a DNA want to
replicate and increase its number.
It's not error-free, so there are some mutations in this DNA structure, and sometimes for example, in two cells in the same tumor, the structure of the DNA is the same, but there are epigenetic mutations. Some of the genes are off, but in other cells, some of the genes are on. Mm-hmm. So as we lose the time, this heterogeneity is getting worse. As
I understood, you mentioned, I want to highlight your comments about the second reason, and you mentioned that by increasing the time, the occurrence of the simultaneous genetic manipulation, I mean that the simultaneous mutations will be increased by time, and so it will be make it more complicated how we can just control the behavior of the tumor cell within the tumor parenchyma. Great, thanks a lot. Daniel, as your classmate mentioned, the early stage detection of the tumor can just increase the survival rate because of different reasons. What's your idea about it?
As my friend mentioned, according to the critical importance of early cancer detection, I want to say that the ability to detect cancers in early stage, even before the symptoms appears, is the most effective public health strategy against this disease, due to three main reasons. First reason is improved survival rates. Cancer caught at localized stages, meaning stage 1 and 2, have shown significantly higher survival rates, about 5 years, in comparison with those who are detected in the other stage, meaning metastatic stage. The second reason is less intensive treatment. Early stage cancers are often treated with less extensive surgery, or less aggressive chemotherapy, or more localized, targeted radiation.
You mean that it can just reduce the economic burden to each patient and even the health system?
That's one of the main reasons.
Great.
And as a result, reducing the side effects and the recovery time for the patient. The last reason that you also mentioned is cost effectiveness. Treating early stage cancers, is generally less complex, and thus it requires less. It is less expensive rather than the other stages. And I want to also mention, among all the detection methods, non-invasive methods are the most appealing due to some reasons. For example, they are quick, they have minimal risk for the complications, and they avoid of patient discomfort. And within non-invasive methods, nowadays, non-invasive biopsy methods, especially exosomes, have shown great promising future due to another three main reasons.
You mentioned that we just monitoring the secretum or the factor released by the tumor cells, which are impacted inside some nanocyte vesicle like exosome, could also help us to just detect the tumor cells at the early stages.
Yes, because of the unique feature, one of the unique features of the exosomes, that they can protect their content, which I'm going to give more elaborate in the future. According to what I mentioned earlier, why exosomes are better option in comparison to other non-invasive liquid biopsies, there are some reasons. The first reason is ultimate non-invasiveness. The ease of self-collection for the patient with removing the risk of a blood draw and the bother of visiting physician office has helped the patients in a way that they can have routine screening.
And you mentioned that a regular monitoring the individuals by some specified time can just give us some chances to just better diagnose the occurrence of some of the tumors and even that some of the cancerous conditions.
Yes, given that the information is updated daily, and this is one of the main advantages. The other detection methods are lack of this ability, especially the surgery methods from tissue. The second feature is a proximity advantage. For cancers that shed their material directly into urinary tract, exosomes provide a highly...
this is some of the tools. So just a monitoring, easy step to monitoring the tools for just detect the cancer patient, even the urine and other biofluids. Yeah.
Yes. As I was saying, these exosomes provide highly specific and concentrated source of tumor-derived biomarkers. And the last reason is stability and information-rich cargo. The protective bilipid shell of the exosomes can protect its content, which include rich variety of DNA, protein, and RNA. And this allows for a deeper, multifaceted molecular analysis of tumor status and function in comparison with, for example, isolated DNA fragments. So, in conclusion, the goal of achieving high rates of non-invasive cancer detection in early stage has supported well by this method. I mean, non-invasive method, biopsy method, meaning using urinary exosomes.
Great. Thanks a lot. I'm going to make some concluding remarks about your comments. As you mentioned, the early stage detection can just increase the survival rate and even that will reduce the economic burden. Your suggestion is that monitoring our biofluids and the tumor cell secretum, even that the biomolecules can enable us to detect these tumors at the early stages, and even this will also make options to selected non-invasive approaches for just detection of the cancerous condition. Thanks a lot. Mrs. Haddadi, right now, at the moment, different clinics and hospitals and laboratories use different modalities for detection of the cancer and even that the tumor cells inside the biological systems. Can you enumerate some of them?
Yes. First of all, I find it very interesting to discuss a comprehensive and advanced methods to detect cancer detection and in cancer detection and diagnosis, with a focus upon pathological, molecular imaging and emerging technology that are shaping precision oncology.
Great.
Cancer diagnosis is no longer limited to identify the malignancy. Instead, it's aimed to characterize tumor biology, behavior, and guide individuality methods and therapy. In my opinion, modern cancer diagnosis is a multidisciplinary and multi-step process that it's related to clinical evaluation, imaging modalities, laboratory tests such as biomarkers, and histopathological and genetic and molecular profilings. Among these, histopathological confirmation is remain gold standard for diagnostic and therapeutic decisions.
Great.
Pathological examination involve the macroscopic analysis of tissue samples obtained through biopsy. Biopsy based on tumor size, location, and even patient condition. And we have common biopsy techniques includes needle biopsy, with, for example, we have fine needle aspiration, FNA, or we have surgery biopsy and endoscopic biopsy.
Great.
Histopathological, of course, evaluation help us to know the lesion is benign or malignant, and we can know the grade of cancer, in which grade, and subtypes, differentiation, and even necrosis or mitotic activity.
Great.
In addition, we have another techniques such as IHC.
It's abbreviated form of the immunohistochemistry.
Yes, that used for detect specific protein markers with using antigen-antibody reactions. For example, we have ER, PR, or HER2 for...
These are some specific markers for the different types of cancers. Great.
Breast cancers.
Great.
Of course, I can mention another techniques such as tumor markers and these things. But I think because the cancer diagnosis is complex, we can't detect with just with one single way. It's, I think, tremendously difficult. We should have multiple disciplinary and...
Great. Thanks for your first comments. According to your comments, as I found it's very interesting, and different modalities and different tools and diagnostic tools have been applied during the past decades to just to diagnose the occurrence of the tumor and the tumor condition inside our biological system. But some of them remain the golden standard as a pathological examination, because we're going to just directly sample the tissue or biofluids from the patient body, and then by making some of the processes, we're going to make it visible to the pathologists. And so we can just monitor the degree of the mutations and even that changes in cell shape, behavior, and even that any reactivity, immune cell reactivity, the occurrence of the other aberrant condition that can just facilitate our diagnosis. Even that in some cases, actually, this method could not just alone help us to just make some consolidated decision how we can just treat and just subject our patient to the non-therapeutic regimes. And so their combination will be essential. Thanks a lot. Very further comments. Mrs. Mamagani, among different modalities and diagnostics and even detection methods have been used for detection of the tumor cells. Monitoring the gene expression and even mutation in genome of the cancer cell can help us to just detect any aberrancy. And because of the, these changes can be occur at early stages. How do you think about it?
Yes, actually, we have different methods for cancer diagnosis. But the most common, the most newest, and also the range in methods has genetic and molecular methods, which we diagnose cancer in 3 steps: DNA-based, RNA-based, and epigenetics levels. I want to start with DNA-based methods. We can divide this method into groups. First one is targeted analysis. In this method, we just only analyze a few genes, and we use Sanger sequencing method.
You mentioned, sorry, I'm going to interrupt. You mentioned that we have some key genes that are integral to occurrence of that specific types of tumors, and by monitoring their expression and even downregulation, we can just predict the occurrence of the cancer.
Yes, that's right. Yeah. Sanger sequencing is a common and most known method in analyze targeted DNA or genes.
Sequences,
yes. By using this method, we can understand different mutations and different alterations in some genes. And second method for targeted DNA sequence analyzing is FISH, or fluorescence in situ hybridization, which is very commonly used for HER2 genes in breast and gastric cancers, because this gene can amplification in many region of chromosomes.
You mentioned that by using the first and tag the complementary sequences we apply to each cells, and these complementary sequences can just directly attach to the, for example, mutated sequences, and so by the reactivity we can just predict the occurrence of that mutation in our target cells. Yeah.
Yes. And second group of DNA-based methods are whole genome sequencing, which we call them next generation sequencing, or NGS. It has 3 platforms. The first platform is only we analyze 50 to 500 cancer associated gene sequences. And second one is a whole exome sequencing. In this platform, we only analyze protein coding genes. It means about 1 to 2% of whole genome. And third platform is whole genome sequencing, or WGS. We analyze all the coding and non-coding sequences in through the cells, and
so this approaches will provide us the bulk information about the whole genomes and exons and inserts, and so by monitoring and even that according to our objective, we can just select some of them, then just predict any pathological condition like a cancer and tumors. Yeah,
that's right. The second method is RNA-based methods. We have 2 common methods, RT-PCR or reverse transcription PCR, to analyze expressions and some mutations in RNA, some specific RNAs, and also RNA sequencing, for mutations, expressions, and also we can use this method for diagnose different types of proteins which only synthesis in cancers. The third methods are epigenetic methods, and the most commonly used in clinical is methylation profiles, and also it's very popular for brain tumors diagnosis. And second method in epigenetics, it's chromatin accessibility assays. It's very good potentially in future diagnosis, but it's now we don't have not more information about this method.
Great. Just by using some of the sophisticated tools and monitoring the subtle changes in the genomes and even that in the gene levels, we can just predict any changes you mentioned in the protein levels, which can just affect the cell behavior. Great. Thanks a lot.